not easy. Always remember, it's not that simple, the same thing. The tryptophan is not simple. They have another thing to control, which is called attenuation activator. I'm not going to go there because it's completely genetics. It's too complicated. Okay. Another example I don't want to mention. It's too complicated. I don't want to become a genetics class. There is another operand you learned in your other classes, maybe biochemistry class, which is combined both positive and negative control, is Albert's operand. You will learn in your other classes. It's a circle. Like a 400-meter stadium running lane. It's going to both can do positive and negative control at the same time. Okay, so that's something else I just want to let you know. My point is that these two persons get together is not easy. They have lots of the factors will control them. Okay, that's why it has become a complicated knowledge for that. Okay, when we finish talking about here, we want to talk a little bit of genomics. It is related to the lactose. Okay, so we want to talk something else. Let me see the time. Okay, we still can talk 15 minutes, so we're going to talk more. So what happened? The story is, I'm going to turn this off. So the story is, once the people find the lactose operand, do some research about it, they're going to be to try to isolate and purify LAC-Z. Because LAC-Z is going to pay much attention during the transcription, translation. They want to purify. So how are they going to do? Okay, they need a, let's say, this is a, we call it a vector, PUC vector. Okay. And you need to know, bacteria sometimes can have some vector. There are the 3 requirements to become a vector. Number 1, a region of replication, which means they can do replication by themselves. Number 2, usually there's a selective marker. And the selective marker, we usually use ampicillin. Because most of the E. coli is sensitive or susceptible to ampicillin. Some of them are going to be resistant. So we have this site called AMPR. Number 3, you're going to have a multiple cloning site. Okay, now how to do that DNA recombination? There is a very magic stuff called restriction enzyme. Okay? Restriction enzyme. The first restriction enzyme happening in the world is ECO-R1. They recognize a very special sequence. Is GAATTC and CTTAAG. What it looks like is that looks like from right to back, left side to right side, it looks like so uniform, is that right? They're called sticky ends. So ECO-R1 can recognize the sticky ends. So we want to talk a little bit about here. What is the restriction enzyme? How people find it? I tell you one thing, it's very interesting. Long time ago, when people find a bacteria has a restriction enzyme, it's not very often. The frequency in the bacteria is very low. It's about 1 per 4,000 cell. May have it. Okay, so they're not very frequent. Now, why they have it? They are actually at the beginning. They are trying to see to protect themselves. Because bacteria, when they're there, some virus will attack it. You call it a bacteriophage. Some bacteriophage can hydrolyze these type of sequences. So for themselves, how they do? If they have the restriction enzyme, they can cut the virus if they have these type of sequences. For themselves, they could be attached with a different methyl group, created a methylase. So, said a very simple restriction enzyme at the beginning found in the E. coli. It is an immune system for bacteria. It is not dummy. You think about, oh, you have the restriction enzyme. How come by yourself you still have the methyl group gonna be there? It's an immune system for the bacteria itself to protect attacking from the phage. Then later on, people isolated and used for recombination of the DNA. Okay, so what happened is, if we do a very simple example, you have this guy right here. Okay? You have G, T, T, A, C. So you have A, T, C, A, T, T, G. This is your sequencing. Okay? This sequencing, this is AMPR. It's a selective marker. This is specifically for LAG-Z, which means galactosidase. If you have a restriction enzyme, you can cut it equal R1. Okay, then go there. Go a new vector. This vector is PUC, let's say. Okay. You go find a new vector. This new vector go here. Have a little bit of new gene. And then gonna carry on. G, T, T, A, C. C, A, T, T, G. We're gonna combine it. Now what's going to happen? This is a new product. This is the original one. Now, of course, recombination not going to always happen. So sometimes you will have these empty vector. And sometimes you will have a vector with LAG-Z. Now, once you recombine, what's going to happen? This LAG-Z is gone. Okay, because you're carrying on the new sequencing right now. So this LAG-Z function is gone. What they do? They're going to plate it on an agar. This agar called X scale. It's a, I will say it's a surrogate of lactose. How do you know the recombination is successful? If you see white colony there, which means the vector is there and the recombine is attached. If you see a blue colony there, which means only vector. Why? Because X scale is a surrogate of lactose. If the vector, only vector is there, or the original sequence is there, the LAG-Z could be hydrolyzed, become galactose. And this is showing you blue color. So very simple. Originally, you have LAG-Z gene. LAG-Z gene can hydrolyze X scale scale is a surrogate of lactose. You can think about this lactose. Become galactose. Showing here is a blue color. Okay? Then, if I have a restriction enzyme to cut it, I add a new sequence there. This flag Z function is gone. They're showing on the agar, scale agar is white colony, which means I successfully recombined. So this is one of the way to do. What are the other way to do? Running a gel electrophoresis. Usually 5% agarose. Okay, it goes here. You have a band right here. You have a band right here. You have a band right here. Okay, this is positive control. This is positive. This is negative. This is positive because DNA is negative. And the small molecular, small BP base running faster. The large one is on the top. There's another one we can separate it. Okay, just very, very brief. I don't want to go too detailed. This tells you people find the lactose output. Find the lac Z. It's very magic there. They try to purify it. Therefore, they do more research. They find the restriction enzyme. The restriction enzyme originally is finding the bacteria is an immune system to protect it by themselves, to attacking by the fudge. Because for itself, it will become merciless and then can protect it. So people do more research. The very typical research is restriction enzymes equal or 1. The special sequence they notice is GTTAAC. Become a sticky ends. And they cut it. They find a new vector, which is PUC, or other vectors, they combine it. Once they combine, the new combination is going to lose the function of lac Z. Therefore, on the X scale agar, which is showing white colony, and the blue colony here, which indicates the X scale has become galactose. So this is the way we can identify it. The other way, using 5% agarose gel electrophoresis. Because DNA is negative charged, and on the top negative, bottom positive, and you go wrong. And you see the small molecule is running fast, small base pair, the BP components is running fast, and the large one is running slow. That's why one is on the top, one is on the bottom, and we can do some of the isolation and identification. Okay, so that's all I have for this section. And on Thursday, we're going to start to do the review, so go ahead to do the review questions.